This is a work in progress, and I will describe only just one part of what we are doing with the team that Thierry coordinates at CIAT. And this, our work within RTB Foods here in Cali is on cooking quality for boiling cassava roots. So as it is often the case for most of our crops, there are a large diversity of factors influencing cooking quality in our products. Genotype, of course, environmental conditions, age of the plant in the case of cassava, duration of the storage until processing, etc. And that leads to a huge experimental error, mysteries, puzzles, and uncertainties that we want to uncover during this project of RTB Foods. The other issue is that there are many factors to consider when we define cooking quality, like cooking time, texture, appearance, and flavor. And please be aware that I am a breeder, not a good food technologist, so I will be using some terms in a loose way, not in the proper way you food technologists use it. But out of all these factors, we are trying to concentrate on cooking time, which usually is estimated by a very simple technology, which is the fork method. We just place the roots into the boiling water, and every once in a while we try to penetrate the root with the fork until adequate softness is achieved. And this is what we call optimum cooking time. However, this method is very subjective and labor intensive. The other thing that we have noticed is that roots that cook well change the angle of the root section. And let me explain what I mean by that. So basically what we do is cut sections of the roots transversally, and then we make a longitudinal cut to have half root sections, as I am indicating here in the photograph in the right, in the left. Do you see the pointer when I talk to you?
Yes.
Yes.
Okay, so
yes.
basically, what will happen is that as the roots boil, if they do well, this angle will be reduced. And here is the way we are now doing this study. We place several root sections in boiling water, and then here you have a root that doesn't cook well because the angle remains more or less around 180 degrees, and another group of 4 sections where the roots start to close like a fan, a hand fan. So this is something that we have correlated to good cooking quality. So what we have done is select a group of 36 clones that are contrasting for cooking quality. And that is not a trivial issue because the environmental factors affecting cooking quality implies that you cannot define good cooking quality just in one harvest season. So we use historical data to select clones that consistently are good or bad for cooking quality. We planted many plants from these clones, and we started harvesting the roots in October through January this year. By the way, these clones, these 36 clones, are the ones that we have used as progenitors for developing biparental populations of crosses between good and bad cooking quality to study heritability and develop markers and produce segregating progenies that we can use in the different activities that the project is doing. So we harvested roots, few roots from each genotype starting in October through January. That means plants that were 8 to 11 months after planting. For each harvest, a detailed study was conducted by Thierry Tran. This is a very detailed study where we got about 36 sections of different roots and randomized to do different studies, which turn around the dry matter content, texture, but the boiled roots were boiled for 30 minutes, for 60 minutes initially, and we stopped the boiling at 5 minutes, 10 minutes, 15 minutes, take them out of the boiling water, let them settle for a few seconds, and then waited. Then we measured the angle of the sections at the end of the cooking, the boiling time, as well as the texture using a texturometer. The important thing here is that softness is related in a paper published about 10 years ago with the weight of the roots. The point is that, as I will say later, when the roots boil, start absorbing water, and therefore the section of the root becomes heavier. What I am going to describe in this presentation is just what we did in December when the plants were at 10 months old. Sorry, I cannot go back. And this is what I'm going to describe. Sorry, I moved ahead. So just to give you an idea, these are the optimum cooking time across the 4 harvests of the 36 genotypes. So this is a variation in cooking time of different roots and harvest times for the 36 genotypes. As you can see, some materials cook very quickly and some materials don't cook at all. This is 60 minutes after cooking, and there is nothing happening there. Incidentally, I think it is interesting to note that in some cases, there is a very consistent behavior of different roots from different harvests of a given genotype, very clear response. Whereas in other cases, you have a large variation. This is a typical variation that we see when we try to assess cooking quality in our crops. Sorry, I will leave the presentation because I wanted to present this. So the other thing that we have seen, this is the average for the 4 harvesting seasons for cooking time. And this is a beautiful example of the impact of environment on cooking quality, in this case, cooking time in cassava roots. Very fluctuating kind of performance. Now, the point is that, as I mentioned earlier report indicated that short cooking time was related to water absorption of roots while boiling. So roots that cook quickly absorb water fast. And if we can take the sections out of the boiling water, they get heavier because the water they are absorbing. Now, our hypothesis is that if roots absorb water while boiling, then their density will decrease gradually as they absorb water. Dry matter content will go down. And then if we weight the roots while they are in the water, the weight should decrease because the density will be reduced. This is a principle of the gravimetric method. So this is what we did. We use a digital scale. The scale has in the base a hook where we attach the boiling waters, the boiling roots hanging from the scale. And it is really fascinating because as soon as you place the roots in the water, in few minutes, the initial weight will start to go down. And this is just a ticking process, and every measurement the scale makes will gradually go down. So basically, what I am going to describe to you is the kind of results that we got. This is a typical performance of one of the clones that we evaluated. Here is a weight in water expressed as percentage of the initial weight, just to overcome, to facilitate the comparison between different roots, because not all the roots weighted the same. So this is percentage of the initial weight. Of course, at time 0, the percentage is 100%. And this is what you can see through the process. And we will analyze different parameters to assess the performance of the roots. And in the plots, you will see the name of the genotype, the optimum cooking time, and the angle of the roots after cooking. So we use 3 parameters.
Yes, Hernan, you have 3 minutes left. Sorry to interrupt.
Yes.
Yes, please.
We use 3, and I have more because I started at 9.5. Anyway, so I use 3 parameters to assess the performance of each genotype. One is the area under the curve. Smaller the area, the faster the roots should get soft. The second is a linear regression coefficient. In the straight line regression, and the more negative the coefficient, the better for the roots to get soft. The third one is the first coefficient in the quadratic regression model. That is the coefficient that we will get, that we are interested in. And the larger this, the more negative this coefficient is, the better. So here you have an example of what we see. Some genotypes do not change density at all, basically. And this performance is related to long cooking time. Basically, these roots did not cook. In this genotype, we have a very consistent regression, just a linear decrease in the density of the roots, and very encouragingly, we have a very fast cooking. And other genotypes, the one that has more of a quadratic kind of performance, like these ones, also have very good cooking time, except the black sheep that always happen in our trials. We always have somebody performing unexpectedly. So, what we then did was relate the optimum cooking time to the different parameters that we have used, taking data after 20 minutes, 30, 40, 50, and 60 minutes of boiling. This is basically a good way to summarize the results. basically the best parameter explaining optimum cooking time is the linear regression coefficient with data up to 30 minutes. Interestingly, if you take more data for longer period of time, you start to see a reduction in the precision because of the exponential or the quadratic performance that many clones show. Finally, if we use this parameter, that is a linear regression coefficient through 30 minutes of boiling with optimum cooking time that goes up to 60 minutes, this is the relationship, and this is the coefficient of determination that is about 0.66. The interesting thing here is that we can make a separation using this arbitrary linear regression coefficient of minus 125, and you separate nicely the ones that cook well versus the ones that don't cook at all. And you have few outliers in the process. So the conclusions are that root lose density as they boil because of the absorption of water. What the Thierry team has done in the detailed study demonstrate that in fact cooking time is related to water absorption. Water changes in density are correlated with optimum cooking time, acknowledging that there are few outliers. So basically, the beauty of this idea is that we can have a relatively high throughput system. This is not truly high throughput, but if you have to assess cooking quality or cooking time in 300 genotypes, the conventional method of the fork is unthinkable. So the idea is to have a battery of 10 weighing stations. Each scale is connected to a computer that automatically downloads the data that each weighing station generates, so that we boil the roots for 30 minutes. And then the other interesting thing here is that we can put 2 kilos of roots, and that will be better because we don't have problems of sampling size. This way we can easily have 100 to 120 genotypes screened in a day. So, and the main objective, because this is not a definite step, is use this approach to pre-select those that cook well. And this is the end of my presentation. Thank you for listening.
Thank you.
Thank you very much, Hernan, for this very instructive presentation. And on this protocol, which could replace the fork method, which is much faster than the fork method, and possibly which can be adapted to other RTV props. So thanks for volunteering for this first webinar. And now feel free to ask questions to Hernan and interact with him.
Hi, this is Thierry from CIAT speaking. I would like to add a comment. Hernan, if you can go back to the previous slide, the one just before the conclusion.
Let me.
Yeah.
I am still sharing the screen.
Yes. Yeah, that's
Here.
one. Yeah. Yeah, as you mentioned, it's a work in progress, and those data come from only one harvest, and we got like two groups of points, two groups of genotypes that are quite well separated. However, when we start to do repetitions with different harvests, we start to have more points that populate the space between the two groups, and things become a bit less easy to separate. So this is something to keep in mind for the optimization of the protocol.
Yeah, I like to highlight that this is actually the most precise way is to do what the Thierry teams is doing, but that is much more labor intensive. So this can be used to screen large populations and then allow you to concentrate on those genotypes that deserve closer attention.
Can I comment on that? Hello, everybody. Happy holidays. I guess then you could do a G by E analysis to clean that up if you end up screening from 3 fields or something.
Actually, what the G by E will explain the variation for cooking time. What we want to develop is something that should not be affected by G by E, and this is this predicting parameter for cooking time. So G by E will affect cooking time, but G by E should not affect, I would expect, the relationship between water absorption while the roots are boiling. There might be some environmental effect, but I would expect that will be minor. I don't know, what do you have to say, Thierry?
Meeting. Yeah. I think what we saw, because this was one harvest. We did 4 in total, and when looking at the water absorption and the increase in weight of the roots in air, we saw differences between the harvests. So there is an environmental effect. Compared to the genetic effect, it was less, maybe 25-30% of the variation.
So basically, the standing hypothesis we have is that cooking time depends on quantity or quality of pectins in the cell walls, correct? If that is the case, then the roots from the harvest at 10 months to 11 months should not change that much for the pectins.
Yeah, this is something we haven't investigated in enough detail yet. What we saw in some genotypes, for example, the pair 183, is a very strong change between 10 months and 11 months. Yeah, at 10 months, it was still very soft, cooking quickly, and very acceptable for consumption. Pair 183 is one of the best clones we have for boiled cassava. And when we came back 1 month later for the following harvest, it was very hard. We could see much more fibers, and it seems that in 1 month that particular genotype changed and increased the amount of fiber they had.
But in this case, did the water absorption change or was the same?
Well, yeah, it reduced a lot.
It reduced a lot.
Yeah.
But that is the point. So the relationship between cooking time and water absorption or changes in root density remains constant.
It remains, yeah.
So what we want is a protocol that can reliably predict optimum cooking time, not a protocol that explains G by E.
Yep.
Okay, so we have...
Hello, this is Bolanli.
Yes, Bolanli, we have 3 persons raising hands, as you can see in the participant list. So we have Didier who raised his hand, and Julien, Dominique, and you, Bolanli. So Didier, if you want to ask a question, please.
Okay.
Oh, yes. Thank you very much, Hernan. My question is in relationship with the bridging program, and I wonder how this how can we link the water absorption and to the physiological basis of cassava if we plan to get molecular marker to use as breeder tool. So I would like to know if you test the relationship between water absorption and conductivity of material at harvest and during the process.
What do you mean by conductivity?
Conductivity is the cell wall properties to is a kind of permeability of cell wall.
Okay, we are not doing this, and it seems a sensible thing to do. What we want to do when we harvest the progenies between crosses of good and bad cooking quality progenitors is to screen them through this kind of approach to get more precise information. So basically, we want to set up this battery, weighting battery of scales to do an initial screening, and then based on the results, perhaps select the populations that are more promising to do more detailed studies, including the
conductivity
study of molecular markers.
Very much.
Yeah. Didier, we have started actually preliminary tests, but then the confinement started, and at the moment the activities are stopped. We haven't been able to see any trend or any relation yet between conductivity and water absorption or cooking time.
I understand that it could be maybe the next step.
Yes.
It's not a priority now. Okay, thank you very much.
Hernan, thank you very much. I have a comment about your high throughput method. I don't think you need to buy 10 weighting equipment and 10 because if you weight the piece of cassava after 30 minutes in air, you will have the same result that the point that you will have at 30 minutes in water. And really, it's better to invest in a battery of pots to cook and waiting exactly at 30 minutes of boiling the piece of cassava. You will have exactly the same result. You don't need to have this dispositive of constant waiting during the cooking, because you will define 30 minutes as your reference, and you will boil 30 minutes the roots, waiting at time 0 and time 30 minutes. And with that result, you will have exactly the same result that by waiting in water or by waiting in air.
I disagree, Dominique, because this is a very reduced number of samples. You will have, say, for instance, here, I am using 35 minutes. You don't see much difference in the relative density of the roots among these 4 genotypes. But this one lost density at slower pace than this one. Okay? It happens, and this has, they have the same cooking time, but the beauty of this system is that you can distinguish between this type of performance versus this type of performance. And the beauty is that we will have data not from few genotypes as it is here, but of 300 genotypes. So at the time being, we don't have evidence that this is different than this, but it will be nice to know if indeed over a large number of samples, this drastic difference between this type of performance and this one has or not an impact. Only time will tell. But by the way, it is. I don't think it is very expensive to set up the battery of scales.
It depends. Maybe you can wait at 10, 20, and 30 minutes if you want. But I think 20 and 30 minutes is enough to see this difference. And if you don't want to buy 10 balance and waiting the system and the central of acquisition, because it's nice to have this course, but it's
the other Dominique, the other advantage is that you don't have a single number. If you make a mistake writing down the numbers or data logging the data, is only a single point. Here you have a continuous line that is certainly more reliable. You have many more data points. But again, this is something that we have basically 8 months to think about.
Yes, thank you.
Thank you. Julien, do you still have a question for Hernan? Then we have
yes.
we had Bolanle and Ismail.
Okay, thanks Hernan and CIAT team. My question is, did you try to correlate this result kinetic water absorption with instrumental texture?
Thierry?
So, sorry, I was reading the chat at the same time.
Thierry, I asked for Hernan, did you try to correlate this result kinetic water absorption with instrumental texture?
Yes. We see the result presented by Hernan, not yet, but the water absorption, so the increase in weight in air, we find some correlation with the gradient at the origin and the area under the curve of the texture measurement. So it's like the more water you absorb, the smaller the area under the curve. That is, the softer the sample.
Okay.
Okay, Bolanle, you can ask your question.
Thank you very much for the very good presentation, Herman. I was just wondering when I saw the setup, and I was wondering, are we going to have that type of setup for each variety? Like if I want to check 10 clones or 10 varieties, will I have to have the 10 setup? Hello?
Yes.
Yeah. Can you hear me?
Yes, we can hear you, but I miss the final questions. 10.
We saw the picture of a setup of the experiment. The picture that you showed us.
Yes.
I'm wondering, if we have 10 varieties, are we going to have 10 setup like that?
Well, this is up to us to decide how we do this.
Yes.
This will not happen. This was something set up as a demonstration during a meeting we had. Things can be organized in a much simpler way.
Okay.
And it doesn't have to be 10 scales. It's the way you want to organize it.
Okay. Thank you, because I was wondering how it will be. Thank you. That's just what I want to be clear. That's what I want to be clear about. Thank you.
It's related to sampling. Like to do one genotype, we can plan to do 2 or 3 replications, laboratory replications. And if you want to do that in parallel, we would need 2 or 3 balances. And then if we put 9, let's say 9 balances in parallel, that means we can do 3 genotypes at the same time and increase the throughput of the method. If we have only 3 balances, we can do 1 genotype at a time, 3 replications per genotype. The test would last 30 minutes, and then after 30 minutes, we move to the second genotype, the third genotype, and so on. So the number of balances is related to the number of genotypes we want to do at the same time, and then the throughputs and how many genotypes per day we can achieve.
Okay, thank you.
Okay, we have a question from Sylvie from Côte d'Ivoire, CNRA, and then I saw 2 questions from Robert Mwanga from Uganda and SIP Uganda, and from Michael Adesokan from IAT in Nigeria. So Sylvie, can you ask your question, please? Sylvie, are you online? Do you hear us?
Perhaps she's mute.
She is mute. Let me. Okay, I don't see her anymore. She's not raising her hand anymore, apparently. Robert Mwanga or Michael Adesokan, do you have questions? Do you want to share your questions with the group, with Hernan directly?
Yes, thank you very much for the good presentation. Now, when you have significant differences in altitude, the genotypes at higher altitude grow slower. That means they will be younger. If you harvest at 10 months, they will be, in time, they will be at the same age. But in growth, they will be younger. And you can even see that by visually. So you adjust for the altitude, for the differences, because the ones at higher altitude grow slower. Do you adjust for that time?
All the materials in this case were grown together at Palmyra. We agree and we publish a paper on the huge impact of altitude on starch quality. So it is likely to be a very different response when we grow the same genotypes in different environments. However, here, and this is a very important statement, we are not interested in analyzing G by E at this moment. We are interested in developing a good system that will allow us to predict cooking time in a facilitated way. And as Thierry demonstrated, even though there were big differences in cooking time in the different harvests, like you see here, these are the average cooking times in the 4 harvests. That will be more or less, if you wish, the impact of growing materials in the coast or in Palmyra. But anyway, the important thing is that the relationship between water absorption or changes in root density and cooking time remain constant or more or less constant in spite of these huge differences in average cooking time. I don't know if I am responding to your question, Robert.
Very well. Thank you.
Thank you. I've seen that the questions from Ismail and Michael has been answered already. Is there any other question from somebody?
I like to add, I find it fascinating because we always knew about the gravimetric approach to estimate dry matter content, which is actually root density. And what is fascinating is that for the first time, you see this in action. It's incredible how quickly the weight in water begins going down, and you see it real time and in a change. So it's the same genotype losing density. It's a very interesting process. So it was like a toy.
Thank you, Hernan. Anyone else? Some more comments or questions? If not, we should probably close this webinar. So I would like to thank you.
The last one. I have one.
Okay.
Now this is really a good progress. And now, as Julien said, we need to try to have some correlation with texture analysis, with sensory analysis, and obviously with NIRS. This is just the beginning of the work. And thank you very much, Hernan, for this presentation. I think it's very, very open-eyed for everybody, and the work is going.
The other thing that is always nice in our case is a good correlation, the good relationship between the field breeding team and the root quality lab. So we have a constant exchange of ideas with Thierry. As a matter of fact, this idea of assessing root density came as we were working with Thierry to the lab. So it's very important that breeders and food technologists spend many hours together fighting, discussing, and wondering about what we get and brainstorming about ideas that we can explore. This is the result of discussions that I had with Thierry, and that is the only way for us to have success.
Thanks, Hernan, for this very, very good message to the group. I think it's what we need to do in each center, and that's why RTB Foods also, that's what RTB Foods are aiming at, to make food scientists and breeders working together more systematically to improve adoption of new RTB crops, new hybrids. So, and I've seen a question regarding this work, if it has been published yet. I think it hasn't been published, right?
As we speak, we are finalizing the manuscript that we will hopefully submit within a few days to the International Journal of Food Science and Technology.